Hello guys, welcome back to my channel. Sorry for not posting any videos for such a long time. It was vacation time, right? The holiday season, so was busy in that. Anyway, so as we were doing carbohydrates and as we completed most of the carbohydrate, the important parts of the carbohydrate. So moving further, now we'll start with a new topic and today we will discuss about DNA, that is the structure of DNA. So, life depends on the ability of cells to store, retrieve, and translate the genetic instruction required to make and maintain a living organism, right? So, this hereditary information is passed from a cell to its daughter cell at a cell division, right? So, when cell is divided, so this hereditary information is passed from a cell, right? And from generation to generation in multicellular organism through the reproductive cells such as eggs and sperm. So the answer to some of these questions began to emerge in 1940s. So when it was discovered from studies in simple fungi that genetic information consists primarily of instruction for making protein. So when there was studied in fungi, so the scientists found that the primary instruction that was in the cell is the instruction for making the protein. So the proteins perform most of the cell's function, as we know that. So they serve as the building blocks for cell structures. They form enzymes that catalyzes the cell chemical reaction. They regulate the activity of genes and they enable cells to move and to communicate with one another. So these are all the protein functions. And we studied the entire protein topic before. So if you want to know more in details about the protein, so you can look at that topic. So, right. The other crucial advance made in the 1940s was the recognition of deoxyribonucleic acid. So the DNA, what's the full form of DNA is? Deoxyribonucleic acid, right? This is the full form of DNA. And it was in the 1940 they found that recognized that DNA was the likely carrier of this genetic information. All right, so we'll see the structure of DNA. So how DNA looks like and its form. So first we'll see the building blocks of the DNA. Building blocks, right, of DNA. So the building blocks of DNA consist of a sugar and the phosphate, right? So for example, like I will make something like this. So let me see if I can use some other color to help distinguish this work. Okay, maybe you can see the difference between brown and this. So this part, the round one is the phosphate. So we'll write it down here. So this is the phosphate part and this is the sugar part, right? And the both together are sugar phosphate, right? So this is a sugar phosphate plus it has a base. So base kind of what we'll make of a base, like maybe like this kind of a box we'll make for this base, right? This is B for the base, and the for example the base is guanine. So this is just for the example. So together that will make one nucleotide, that is, like we'll use this here. Right, so this is the one nucleotide, right? So the molecule of the DNA consists of two polynucleotide chains. So it has a two polynucleotide chain, and each chain or strand is composed of four type of nucleotide subunits, right? The two strands are held together by a hydrogen bond between the base of portion of the nucleotide. So this is just a single nucleotide that I showed you now, right? So this is a single nucleotide, and so from this single nucleotide, so we will make a polynucleotide chain for the DNA, right? So for example, like a polynucleotide, right? Or the DNA strand, that is. So for example, this will make a curve here. Why will make a curve here? So because, so we'll use this one. Okay. So this is like this, right? So first is we have this guanine. I will put G for it, right? Then the second one, the second sugar and phosphate connects together. Oops. All right. And for example, this is cytosine, right? Then another connect, right? Right. This is adenine, right? Oops. I'll put it with the black. And it's thymine, right? So this is like this. So this is connected like this. The nucleotides are covalently linked together. So this is like guanine. So this is the same thing as for the cytosine. So it has a sugar phosphate, it has the base, that is the cytosine, right? And this together is connected with each other by a covalent bond. All right, and this makes the backbone. This sugar phosphate make the backbone of the DNA, right? Right, so this is a just an example of DNA strand, the one. So like the actual DNA is double stranded, right? So this is a single strand, so we will make it double stranded DNA, right? For example, now we'll try to make just in a straight or a linear form. For example, let's use this again. All right, so we'll use this, and this is for example C cytosine, and that is connected with, we'll have to make the big one so you will know the difference between, and this is guanine, so C is connected with the G, and oops, this will make one small one. Nice. So for example, this is like, I'll put it here, A and T. So adenine is connected with thymine, right? I think this is the top is good. Right? This is the same thing, DNA, thymine, adenine. So it's like this. I hope you understand my point. I'm not such a good artist in drawing things. Try best. Maybe that will make a sense, or I'll make a bigger picture for you. That will clearly make a sense. Maybe this much bigger. this is C and G. For example, this is A and T. Alright, you're getting my point. So this is something like this, right? G and C or A and T, and it goes on. Oops. Alright, a kind of this structure is represented, and this is actually, so I did it in the wrong way, because this strand and this strand, I will explain that. So both run in an anti-parallel way in the polarity form. So for example, like this polarity goes on this side, and this polarity should goes on this side. So this should be like this. So this should be like reverse instead of this. Oops. So this would be like here, and it should be like this. It should have here for this, and it does not. It should not have anything here. So in terms of polarity, so all this is, so this is just a linear strand of the DNA, right? For example, this is thymine and adenine, right, like this. So this is how DNA is formed, right? So it has a sugar and a phosphate base and plus the base, so that will make a nucleotide, and this nucleotide is connected like this together to make a polynucleotide or DNA strand, and this was a single DNA strand, and this is how these two strands are connected together with the hydrogen bond, but it is not like a linear form. It is actually connected in a double helix, right? So that's how to make a diagram for it. I will give a try for the double helix. Maybe if I can do that, make a double helix for you guys. Something like this, right? So for example, this is okay, brown. We'll use the brown one to make it. This is, for example, G. I'm sorry, this is connected with C, right? So we'll have here, so if this has here and here, right? Kind of A and T, right? And it has here, here, again G and C or C and G, right? So this is something like this goes in the helical form. Right? Something like I don't think that's a good helix, but but I hope you got my point. Well, anyways, right? So it is not a linear form. It is like this, and this is connected like this together, the nucleotides with the hydrogen bonds. All right. So this is the structure of DNA, right? So a molecule of DNA consists of two long polynucleotide chain like this. These are two long polynucleotide chain, and each chain is composed of four types of nucleotide subunits, right? And the two chain are held together by the hydrogen bond. So this is, I'm sorry, so this is hydrogen bond that is attaching it together. All right. So the nucleotide, this nucleotide are composed of a nitrogen base. So there is one nitrogen base. This we studied already in protein, but I'm just writing it down to refresh your memory. So nucleotide contain what it contains. A nitrogen base, right? And 5 carbon sugar, 5 carbon sugar, to which one or more phosphate groups are attached, to which one or more phosphate groups are attached, right? So this is a nucleotide, right? So nucleotide, what it has a nitrogen base. Like for example, this is the guanine is a nitrogen base. It has a 5 carbon sugar, and this 5 carbon sugar in DNA is deoxyribose. Sorry. So this 5 carbon sugar in DNA is deoxyribose, right? That's why it has a name, deoxyribonucleic acid, right? For the DNA, and the four and the bases, the 4 bases, which are the 4 bases for this. So one is cytose, right? So the first one is cytosine, right? Then the one we saw, guanine, right? Thymine, then adenine. And this all represented with a symbol for this. This is T, this is G, and this is C, right? So these are the 4 bases of nucleotides. So as I mentioned about the directionality or polarity. So this is commonly known as 3. It's written like this, 3 and 5. So 3 is a prime end and 5 is another prime end. And what does that mean? I'll explain to you. For example, here, so 3 prime end, right? So in a simple way, the sugar in the DNA is 2-deoxyribose, right, which is a pentose or a 5 carbon sugar. So the sugars are joined to the phosphate by phosphate group that forms the phosphodiester. So the sugar and the phosphate, how they are joined? They are joined by phosphodiester bonds between the third and the fifth carbon atoms of adjacent sugar rings. And that's why, so the one that is attached at the third is known as a 3 prime end, and the one that is attached at the fifth carbon is known as 5 prime end. So the prime symbol, this one, being used to distinguish this carbon atoms from those of the bases to which deoxyribose forms a glycosidic bond. So therefore, any DNA strand normally has one end at which there is a phosphate group attached to the fifth carbon of a ribose, and the another end at which there is a free hydroxyl group attached to the third carbon of a ribose. So 3 hydroxyl and 5 hydroxyl as well, it's known as. So I will show you. So I will draw something for you. Let me remove this and make this thing half. Alright, I will draw here. Make a line. Okay. This might be enough space. Maybe. Alright, so I'm drawing this a nitrogen base here. So just imagine. So my drawing is not that perfect, but this is one nitrogen base, for example, thiamine, right? And this, connect this nitrogen base. So this nitrogen base, right, as we can see here, this nitrogen base is connected to the sugar and the phosphate, and the sugar is a 5 carbon sugar. So oxygen 1, I think this is correct, right? Oops. For something like this, it's, my drawing is not that perfect. It should connect. Okay, wait. I have to connect that. It's so far away. Let me connect this here. Right, so 5 carbon sugar, and this is in turn connected with the phosphate group, right? So I'll make a simple phosphate group. So it has connected like this with the oxygen. Right? This is P for phosphate. All right. Now this thiamine, for example, this thiamine is connected to adenine, as you can see here. So we'll connect the adenine to it as well. And see, so this has the bonding. Oops, it should be reverse bonding. CH2. Alright, but right now I'm not numbering right now. I'm just drawing the diagram and then I will number it and show you how it is connected. CH2 OH. Alright, this is hydroxyl group. And I think that will run out of space. Right, so again 5 carbon sugar. Maybe this is kind of right. Right, this is connected to adenine, right, H2N, something like this. All right, and this goes again with the phosphate. And that connects again, but the same thing goes on and on. All right, so I will number the carbon for you. So now this is a nitrogen base, as you can see, that's thiamine, right, T. So this base is connected to a 5 carbon sugar. So 1, 2, 3, 4, and 5 carbon sugar, right? So this is a 5 carbon sugar, and this is here the 5th carbon, for example. As you can see, this is a 5th carbon, this one right here, and this one, this phosphate that is connected to it, it's the 3rd carbon. All right, and the same thing here, this is a 5th carbon, and this is a 3rd carbon sugar. Right, so this adenine and thiamine are connected like this with the hydrogen bond, right? And this this bond with the phosphate and the deoxyribose is the phosphodiester bond. All right. All right. So I will just show you a simple diagram here for this so you will understand how this is actually works. Oops. Right. So I'm drawing the 5 carbon sugar. Oh, it has an hydroxyl group, right? And this in turn is connected to the phosphate, right? So it is also a hydroxyl group and this is connected together. All right, so this is the 5th carbon that is outside here. This here is the 4th one. This here is the 3rd one. This here is the 2nd one and this here is the 1st one. This is how 5 carbon atoms are arranged. So this is a base, right, with the base that connects at the 1st carbon, like this, right? So this is the same thing, but I draw it small to show you. So this, for example, here you can see, so this 5th carbon is connected to nothing right now. So the 5 prime end, that is, so that designate, so this is the 3 and there is one is 5 prime and oops, right? So the 5 prime end designates the end of the, this is how it's pronounced, the 5 prime end designates the end of the DNA or even the RNA strand that has the 5th carbon in the sugar ring, right? This is the 5th carbon in the sugar ring of deoxyribose, or it can be just ribose at its terminus, right? So this is at its terminus. It has, so for example, so I was wrong. So this is like opposite 3 and 5, and this is 5 and 3. So these are the DNA strands. So for this double helix, 3 and 5, right? The same as this. So, in a simple way, so this 5 end that is here, that designates that the DNA strand or the RNA strand has the 5th carbon in the sugar ring at its terminus. So this is like, if you can see this part here, so this part is free of phosphate group attached to the 5th end that permits the ligation of 2 nucleotides, that is the covalent binding. So this 5th is a part where 2 nucleotides bind together, 5 sugar. The second one, as you can see, this here, it binds together with the adenine and thymine by covalent bonding, and this forms the phosphodiester bonds as well, right? The same as for the 5th as it's the same for the 3rd as well. So for example, this 3 is ending here, so 3 will have, so 3 will have a free hydroxyl group, right? So the 3 prime end of a strand is named because due to its terminating hydroxyl group of the 3rd carbon in the sugar ring, and that's why it's known also as a tail end, right? So the 3 hydroxyl is necessary in the synthesis of nucleic acid molecules, and it is joined to the 5 phosphate to of a separate nucleotide for allowing the formation of strands of linked nucleotides, right? So like I didn't show, so this you can see, so this 3 allows to join with the 5 through the phosphodiester bond, right? So why I did explain, so because of this, the double helix structure of DNA, just to show that that the DNA strand normally has one end, right, as we can see, at which there is a phosphate group, like this phosphate group, right, attached to the fifth carbon of ribose, sorry, and another end. So this is the three end, right? For example, fifth and the three end, at which there is a free hydroxyl group attached to the third carbon of a ribose, right? So the orientation of this 3 and 5 carbons along the sugar phosphate backbone contains directionality, or also sometimes called as polarity, to each DNA strands. Right, so this is just supposed to show the directionality or the polarity of the DNA strands, right? The 3 and the 5, as I explained here, right? And all the bases, so this adenine cyclozyme, these are all the bases, are therefore on the inside of the double helix, right? They are on the inside of the double helix. The bases do not pair randomly. However, A always pair with T. Right. A always pair with T, and G always pair with C. Right? A always pair with T and G always pair with C. Alright, so that was DNA. So basically, like in this double helix structure, so when this helical turns, so there is like I think around 10 pairs are present in the turn, right? And information is encoded in order or sequence of the nucleotides along each DNA. So each base A, C, G, T or G can be considered a letter or a four-letter alphabet that is used to spell out, right, biological messages. So organisms differ from one another because their respective DNA molecules have different nucleotide sequences. So for example, if I have like a CA GT GTCA, you will have something like AGTC or something, some different sequence, right? This is how it is separated, right? And this is how there are different biological messages that are generated, and we'll study in details. All right, but this was just a structure of DNA that we studied, and in the next topic we'll study the chromosomes. Right guys, thank you guys for watching, and don't forget to